So I think we're recording now. This is the Monday evening session at just after 8. And I mentioned already earlier on that sometimes these do run a little longer, but I'll try to be very efficient and respect your time. So on this session, we're going to have a look through mainly what's going to come up in the exam. So really our main focus will be on the study guide for the exam 1. So if you hadn't had a chance, you will find it, to access it, you will find it either, you can go and download it or print it or just have it on the side on the screen from this week's learning module. And I think that I've already published all study guides for each exam module. So some students prefer to use those even during the chapter that they're studying. So even before the exam is taking place. Some prefer to use it as a tool while you're studying for the exam. These study guides are a little bit of creative in a sense that there are gaps in those. And those gaps are either important concepts or important terms or something that I really want you to pay attention to. So only place where you will get answers to those if they don't make any sense to you is during one of these review sessions. And again, I'm going to just jump back to the my view of the Canvas course, and you should have a similar view when you access this week's material. There you will find on the lecture study resources, the study guide for the exam 1. So this is what I'm referring. It will open here on the screen, but you can also download it by clicking on the blue link. And all of these gaps are important things, and we'll cover those on these sessions. I don't upload a completed PDF anywhere, so it's either by attending to one of these sessions or then watching a video where you'll get the answers. The reason for that is that these study guides contain every single question that you'll see in the exam. So that's why I think that since it's an open book exam, it would be a little bit too much of a help if I would also upload that as it is there. So at least this way, there's a certain element of proactivity that's required for getting the answers in front of you. So the other thing before we get any further that I wanted to mention, and I didn't remember to mention in the morning session. So this is although the sessions are roughly the same, there's always a little bit of difference that I really wanted to highlight was some of the hints and tips that I put together this morning that what you might want to consider before the exam and during the exam and after the exam. These don't apply only to the practical. They're also totally applicable to the full exam that we have. But make sure that you have that you're mindful about the time. You have about 1 minute per question, both in the practical and in the exam. So a lot of the time, these are questions that you either know or you do not know. So kind of going back and forth between the options is usually not necessary. So if you find yourself spending multiple minutes on one question, at that point, I would advise that make a guess, jot down on your notes that this was something that you want to come back to if you have more time, and then move on to the next one. It's better to have at least some answer there, because if it's a multiple choice question, you at least have, based on the probability, chances of getting something right rather than not answering at all. And you can always come back to the questions. There is no negative marking, so there's no loss by just simply guessing. By pure chance, you should get some of those points. If it is the practical, then just write something that's related to that thing. And you can come back and change it later on. But as long as you give me something, I can at least give you partial points. I really encourage you to make sure that you do work through all of the questions. That's the key to getting the full points. And for the practical, I keep mentioning this, that notice that there is no issues with errors in the spelling. I will go through each and every student's each and every answer. I think I go through them 3 times blindly without knowing what student answered what, and are blinded from that information. So you will be getting points even if the spelling isn't perfect. So that's where the partial points step in as well. And do stay calm. Once you have the final version, I will send you a message that this is your that the final grades are up. So the initial grade that you get based on the automated grading might not be the final grade that you're ending up with. And do remember, there's no curveballs. These are as simple as I've told you. So there is nothing to worry about in terms of that. I'm going to highlight, I don't think this is really relating to any of you. Usually this is related to the people who don't attend to these sessions or look at the videos. But if I were to find that the questions are circulating online, that would be a problem for all of us. So I ask you to kind of play nice. I try to make this course nice for you and doable for you. And I hope that you work with me on making sure that I don't have to write new questions for every exam. So I'm going to jump now back to our PowerPoint. Sorry about going back and forth. And just a couple of things that I want to mention for this week. This week, you really, once you have finished the exam, we have reached almost the 1/4 of the course material covered. So once you have points from that, it's not quite 1/4 of the final points because there are some more points available at the very last week, the finals week of the course, but you have already covered a bunch by the end of this week. So give yourself a little bit of a congratulations or slack for that. And obviously that's supposed to be exam 2 there. I don't know. For some reason copy pasted that. Another thing that you'll notice from this schedule is that for this exam, you only have 2 chapters to cover. So for that reason, for example, the lab practical on this week will be much shorter than any other lab practical that you'll see in the future weeks. Also, the amount of material that we're covering in terms of the chapters is a little shorter. We will go a little bit deeper than on the other chapters in relation to the material because we have just a little less material to go through. So that's just something that might help you to kind of put the timeline on a focus that what we are gonna cover. There are no new assignments this week, so spend your time working on the exam and lab practical. And if you have any assignments that you have not completed from the previous weeks, this would be a good time to get those done. For doing that, you might need to get me to reopen or give you an extension of something. If that's the case, message me and be sure to include the details of what assignment it was. If I get a message that's just like, oh, I didn't get it done, the work that I've not yet done, that becomes very difficult for me to go through every single piece of work that you've done. I usually am able to do that, but it might take a little longer to hear back from me. So I encourage, if you include a little bit of detail, I'll definitely get those assignments open for you. The secret to succeeding on these online courses is really collecting all of those points throughout the semester. I really want you to get those. Once you get those, you have way less to worry at the end of the course of how to gather all the points that you need for a certain grade letter. This week, so I'm asking you to do the exam 1 and also do the lab practical 1. Both are due at the end of the week. So on the Sunday, which I believe is the 14th, right a minute before the midnight. And if you look at the point distribution, you'll notice that the actual exam 1 is worth of 100 points. And the importance of that probably makes sense when you refer back to the amount of points that are available on the weekly basis. So really skipping an exam puts you in a huge disadvantage in terms of being able to get the points that you might need for the final grade that you're after. So that was 100 points for the exam, and half of that, so 50 points for the lab practical. What I've done to try to make your life a little easier is that I put together that study guide, which we will be working through on this session. That's going to be our main focus. In addition, I received a request from students about how would you be best prepared for the lab practical. Well, for the lab practical, you've really covered the material while you've done those practice anatomy assignments as part of the coursework. But just when you're preparing for the exam, you'll also notice that there's a practice version of that lab practical. And some students find it useful to go through that. It doesn't put any extra points towards your final score or towards your assignments. But what it does, it pulls questions from a larger pool. And if you go through it once or twice, depending on which lab practical we're looking, there's a little less questions on the pool on this first one. Later on the course, there's more questions because there's more material that has been covered for those practical exams. But if you go through that couple of times, you'll end up seeing every single question that we might ask from you on the actual lab practical. So that might be a good idea to do that. And remember again, also the lab practicals are an open book test. So some students take screenshots, some take their own notes, some have textbook next to them, some do all of the above. So whatever makes you feel comfortable that you're totally prepared for that lab practical. And I probably would encourage you to again think of it in a way that you have about 1 minute per question. Usually it's really obvious. Either you know the structure that you're shown a picture of or you don't know. The question can be something else than just simply naming the structure, but most of the time it is just simply identifying things. In terms of how do you identify, remember that there's going to be that text box. It's not going to be a list of options that you choose and drag where it should go. Simply instead, there will be a text box where you will type in the answer, whatever you think that structure is. And this is where the spelling might step in, that if you make a spelling error, the computer automatically will grade it as a wrong answer. But I will then go back later on and change it so that you get points for that. I also received a question or a request, I think that last semester, from one student that they would find that it would help if you had lab handout or had handouts of the lecture material. So I can't give you the exact same lecture material slides that I use, mainly because those are done by me and there's a whole copyright issue where we might end up otherwise. So what I've done, I've gotten slides that are very, very similar to what I've used from your publisher, and they are there as handouts available for you to download as well. And I think that they're for every single chapter. If not, I think there might be at the very end, 1 or 2 that I've missed. I will keep working on uploading those as we move through the course. And this is the point where I would like us to jump to the study guide. So if you have had a chance to either print or pull up the study guide, that would be ideal so that you see it as we go through. And let me just open it here as well. So you should be able to see now a PDF reader screen. That's mine. And on that, a window, or on that, a document, which is the study guide with the actual answers to all those gaps that we were missing. So we're going to use that to go through the material. And at any stage, if I'm going too fast or if it's unclear, again, shout out or type to the chat. If I don't notice you at the chat, shout out louder and I'll try to stop and re-explain if something doesn't make sense or is unclear. I hope that this amount of zooming in is visible for you. If it's not, you just let me know and I'll zoom in even more. So let's get to the actual material. So for the first chapter, we were looking at the endocrine system. And endocrine system is, of course, all about the hormones. So we're looking at things such as production of hormones, but also how those hormones are released, how they travel throughout the body, how they act, how they are either deactivated or eliminated from the body as well. So one thing that you'll notice in most chapters, and especially on the study guides that I'll end up doing, I try to make some connections between the various systems that we're looking at. And since we are focusing on this chapter, on this endocrine system, I want to just remind you there's about 11 body systems, depending on a little bit of the way how we classify them. Sometimes some prefer to separate cardiovascular system into a couple of different systems. Some prefer to have it as one. Same goes for the reproductive systems and so on. But at large, about 11 body systems. And what we'll notice that the endocrine system really affects all of the other body systems. So the hormones have an effect on all other parts of the body. And we see the reverse, that many of the other body systems produce hormones. So they also affect the endocrine system, or are involved with the endocrine system in that sense. So these all really have a lot of overlap. And this is where it came when I said that it's sometimes hard to really fully understand the endocrine system until you understand all other systems. So don't feel that if this felt like the hardest chapter ever, that you're wrong. It very well might be that it was because you don't have all the information yet. The other thing that I wanted to highlight, kind of getting us to think about the endocrine system, is that we've talked earlier on about the nervous system. And nervous system is another example of a control system or a signaling system in the body. And we remember you finished the nervous system, I think at the end of the BIO201. One thing that you saw about the nervous system is that it sent always very fast electrical impulses, and these electrical impulses were very short lasting. So when the action potential has passed through a neuron, then it's gone. There's not a constant action potential present. This is different when it comes to the endocrine system. Since we are not using electrical signals, but we're using actually those hormones, the hormones take a longer time to travel. However, the benefit of this is that these signals are going to be present for a much longer time in the body. So that's the big difference between these two. So I think that sets the scene quite nicely. So there are similarities, but there are also differences. Another thing that I want to mention, that rarely do we see the endocrine system acting fully separate from the nervous system. Often there's an overlap between the two. Even if you think back, how do the messages pass from one neuron to the next neuron? In between, there was no physical connection, and it was actually neurotransmitters that passed over that gap. Well, we could say that those chemicals, those neurotransmitters, could qualify as endocrines. So there is similarities. Also, many nervous system processes result in certain hormones being released. So see that although we address these separately and we highlight the differences, there is also interaction between the two. kind of also still setting our scene for the endocrine system. When we talk about the field of science where we study endocrine system, we talk about endocrinology. So that's the field of studying hormones, studying endocrine organs that produce and organs that receive hormones, and how these processes take place. Also the deactivation and elimination of the hormones as well. And we noticed that there were many different functions for the endocrine system, and we'll talk about that in a second. But from this list, I really want to highlight this part, that it is through the blood that most of the endocrine system works. So it is within the bloodstream where we release these hormones and how these hormones travel throughout the body. Another theme that I just want to quickly mention, not go into great detail, is the difference between endocrine and exocrine glands. So you remember that glands by large could be divided into these two groups in the body. And of course, we are going to be focusing on these endocrine glands. But to understand what's the difference between these two helps us to understand what's the nature of the endocrine glands then. Well, let's start, though, by looking at the exocrine glands. So where we might have seen exocrine glands in the past is, for example, when you think of the glands such as, I didn't mean to do that, such as sweat glands. That would be a good example. So we know that we have a lot of sweat glands within the skin, or we have lacrimal gland just next to the eye, or we will cover actually three different salivary glands and types of salivary glands later on this course. All of these have a very clear, distinct gland part, so where these secretions are produced. And once these secretions have been produced, they do get released, but they get released through a duct. And that duct takes these secretions to the target location where these secretions will act. So a couple of important things to notice about exocrine glands. They work through ducts, and secretions are released via these ducts, so they can't travel for too far, so they're very locally acting glands. For example, when we look at the salivary glands, we'll notice that there's distinct parts within the oral cavity where each of these ducts opens, so they don't just dump contents roughly somewhere. It's very specific. Well, that's the big difference between the endocrine glands. So let's have a look. How does this business work with the endocrine glands then? Well, again, we do have a gland, and this gland produces certain secretions. But unlike what we saw earlier on, these secretions are now released to the bloodstream. So there's no specific duct, no specific location where they get released. They're simply within the bloodstream. And this bloodstream then takes them to any part of the body where the blood may possibly flow. So hormones actually end up going everywhere. And it is only within certain cells where these hormones trigger responses. And that's because we have specific receptors on those particular cells. So endocrine glands, secretions travel through the bloodstream to all parts throughout the body. So they're not local like our exocrine glands were. So I hope that helps to kind of highlight the difference between the two and kind of set the scene for understanding why endocrine system is a little bit more complex and why we need to consider a few different aspects of that. And there's a bunch of examples that we have already discussed in this when we looked at this chapter, and we will go through those again. There's one specific gland that I wanted to highlight here, and usually we as anatomists and physiologists love things when they are a little odd or when they're off from normal. Well, pancreas is a good example of a gland that is actually both exocrine and endocrine. So endocrine function of the pancreas is what we're going to be focusing on, and that's all to do with the blood sugar level regulation. Whereas the exocrine function, which is the production of the pancreatic juice, which is all about digestion, it's something that we will be looking at later on this course. And we'll end up finding that pancreatic juice is actually the most complete digestive juice that we find in the entire body. So that is efficient secretion. But we are looking at this chapter simply to the endocrine system. And just up, if you're getting feedback, it might be worth checking that do you happen to have a microphone on. So now that we've done the biggest kind of getting us set up, what we will be doing, we will go through all of this handout which we completed together during our first live weekly online session. And if you didn't have a chance to complete that, I encourage you to do so. You will find me discussing with you step by step each of these glands on the week 2 live weekly online session recording. But how we will cover these instead is a little bit of in a different format, but the key points will be covered still on this review session. So let's move on and start with hypothalamus. And the reason why we start with the hypothalamus is that it's considered as the master gland. So hypothalamus controls majority of the other endocrine glands. So often for that reason, we've given it this kind of a nickname that it is the in charge of all of the endocrine system. And if we do a quick diagram, I'm doing here a cross section through the brain. Here's going to be the anterior part, so the front, and here's going to be the posterior part. So we have our hypothalamus within the brain here, and from the hypothalamus we have this little streak that comes out, and this streak is known as infundibulum. So infundibulum is this part that connects the hypothalamus to the pituitary gland. And what we're going to do next, we're going to have a look of the pituitary glands. And pituitary glands actually have a little bit more going on there because pituitary gland has actually 2 different lobes. So we're going to be looking both at the posterior and anterior lobe of the pituitary gland. And there's very different hormones that are released from these different parts or different lobes of the pituitary. What is probably the most important part when we're trying to make sense of these and how the messages pass through is to notice that it is through neural signals that messages travel to the hypothalamus, and from the hypothalamus, messages travel typically always to the posterior pituitary. And if we have further hormones that get released from the anterior, it then travels from the posterior to the anterior lobe of the pituitary gland. So there's almost this defined order of how the messages travel at large. So let's start with the posterior lobe of the pituitary gland. And what you'll notice here that I've highlighted that it can be both neural signals, so signals that are simply action potentials, from the hypothalamus to the posterior lobe through the infundibulum, but there also can be endocrine signals. So they can be hormonal signals that travel only this short distance from the hypothalamus to the posterior lobe of the pituitary. But the two hormones that I want us to focus on in terms of the posterior lobe are going to be oxytocin and ADH. And ADH stands for antidiuretic hormone. So we'll talk about that in a second. But let's start with the oxytocin. So oxytocin is a hormone that has an important role in the childbirth. So on the delivery of the baby. And the other role that it has, and we'll end up noticing that this same process has other hormones that affect it as well, is the milk production and release especially. So both of these processes are a little unusual for what we've seen in the past because they are examples of positive feedback in the homeostatic regulation loop. And we'll talk about what that means. What does the positive feedback mean in a little bit, because it is such an important concept. But you'll notice and you'll remember that most of the time the feedback that we get is negative. So on the homeostasis and regulation loop of that. But remember, oxytocin related to the childbirth and lactation. Well, if you think about that we're giving birth to the child and the baby is going to come out of there, we are not going to stop that process halfway through. So there is not going to be any hormone that gets released in response to giving birth that would signal that, hey, I've changed my mind. Let's suck it in or leave it hanging out halfway. Same goes for the lactation. So having the child pushing against the uterus wall just increases those signals that we now have stimulus here, and that increases the process of giving birth for the uterine wall lining muscles to start contracting. The same goes for the lactation. When the child is suckling on the nipple, that triggers the release of the milk, and that in turn then secretes more oxytocin, and again more milk is released. So it's almost like a cycle that promotes itself. So that's what we're meaning with the term positive feedback. The second hormone from the posterior lobe was our ADH. And ADH does really what it says in the tin. So it has all to do with production of the urine, and since it's anti-diuretic hormone, it makes the urination lesser. So it prevents or decreases the process and the amount of urine that is produced. And we'll actually end up noticing, we'll talk a bit about the ADH in our chapter where we look at the kidney function later on this course. So this is a hormone that I want you to be aware of. You don't need to know exactly how it works. You just need to know where it works and what it does. And exactly how it works we'll cover later. So that's a good example of why a lot of these hormones are kind of tricky to study at this level, because you don't get the full explanation. Well, the next one that I want to have a look is going to be the anterior lobe of the pituitary gland. So now we're focusing on this lobe here. And there we're going to have a bunch of, in the large, I've grouped them as 5 different groups of hormones. What I want you to notice, though, is that all of these anterior lobe hormones, that the release of those, the signal passes through the posterior gland first, or posterior lobe of the pituitary gland. So let's have a look at the hormones that we should know from the posterior posterior lobe. And we're going to start with the growth hormone. So growth hormone really does what it says on the tin again, so often abbreviated as GH, and that refers to the fact that it is what promotes the growth. So we have a lot of growth hormone present right after the baby has been born during the early development. And then the amounts of the growth hormone decrease a little bit until there's another huge surge of growth hormones during the puberty. So again, we see huge amount of growth during the time of the puberty. So that's what the growth hormone does. It physically promotes the growth. You can see misuse of growth hormones, for example, when people are taking steroids in terms of doping for athletic purposes. We can have growth hormones injected to, for example, cattle to get more meat, bigger meat. More muscle mass to those, all sorts of things like that. And those approaches would be generally kind of frowned upon. We don't consider that they're very good practices. But again, there can be disorders. That's why we're learning about normal things, so you can then piece together what happens if there's issues. If we notice that someone's puberty, for example, doesn't start at all, if they are not growing, they're not getting all of those changes that we expect during the puberty, we can have a look, are their growth hormone levels what they should be? And if not, it might be necessary to give them artificial growth hormones. So just putting that out there, we don't go into disorders a lot on this course, but giving you an idea, why do we need to know the normal so you can later on look at the abnormal. The second hormone that we noticed that was secreted from the posterior lobe of the pituitary gland was thyroid stimulating hormone. So again, it does exactly what the name proposes, abbreviated as TSH. So it stimulates the thyroid gland to then release thyroid hormone. And we'll talk about the thyroid gland a little bit later. So you'll notice that, again, this oversight is true both hypothalamus and the pituitary gland in relation to the other glands that we'll see. The third one that I want to mention, again, similar business going on there. We have the adrenal gland hormone. So this promotes the adrenal gland to release its hormones, mainly adrenaline. There are a few other important hormones associated with the adrenal gland, though. So we'll have a look at that a little later as well. Two hormones that I've grouped together here, so if you wish fourth and fifth, are gonna be follicle stimulating hormone and luteinizing hormone. These two hormones are especially relevant to us when we look at the regulation of especially female menstrual cycle. And that's going to be the last two chapters of this course. So again, I just simply want you to have an idea that there are those two hormones, follicle stimulating hormone and luteinizing hormone, and they play a role in the regulation of menstrual cycle in females. That's all you need to know. You can then fill in the gaps once we get to that actual chapter. But that's the reason why we're covering them already at this point, so you know that they would come from the anterior lobe of the pituitary. And the last one is kind of something that's very close to what we've seen before. Prolactin, again, acts on the lactation. So we saw the lactation earlier on as being controlled by oxytocin. Well, there's another hormone, and that's going to be the prolactin that contributes to that as well. So you'll see that rarely is, if it's an important process, rarely are we dependent on only one single hormone. Often, if it's something that's important for us, we have few different hormones that act on that or add to that. So those were the hormones of the pituitary gland. I know I'm going through them a little fast, so if at any point it seems like I'm going through them too fast, give me a shout out. I'm definitely not trying to confuse anyone as a result of this. What I want to do next, I want to have a look of the adrenal gland with you. And what we'll notice, or what we'll remember from the adrenal gland, was that it was located on top of the kidneys. So kidneys, if we try to think about what do kidneys look like, we'll talk much more about kidneys later on, but roughly you can consider kidneys as these kind of a bean-shaped structures. There's a tube coming out of them. The right kidney is a little lower because on top of that we have the liver that kind of takes that space where it would have otherwise been. And we remember that the main function of the kidneys was the blood pressure regulation, also production of the urine. So adrenal glands are located on top of these structures, and they're a little bit like a pyramid-shaped structures. I'm drawing them here out of the scale, so that's not totally realistic what I'm showing there, but I'm really trying to just illustrate that they are present. And when we're talking about the adrenal gland hormones, we actually have to look more inside or to the cross section of the adrenal gland. And what we'll notice that there's two distinct parts to the adrenal gland. We have the cortex, which is the outermost layer. So you've heard the term cortex being used earlier on when you discussed about the brain and how the outermost layer of the brain looked like this sausagey structure, and it was actually gray in color. So that was the cortex of the brain. Similar on adrenal gland, we have a cortex, which is this outermost layer of this gland. And then inside part is known as medulla. That's the center part of the adrenal gland. So that's what I wanted to first make clear or get us all on the same page. So then when we start looking at different hormones, we can make a difference from which part of the adrenal gland these hormones are from. And let's start with the cortex of the adrenal gland. There's actually 25 different hormones that are secreted from there. We're not going to go through all of them. I'm wanting us to focus on two groups of hormones. So this includes a bunch of different hormones that I've grouped together so that we only have two that we need to be comfortable with for the adrenal cortex hormones. So the first one is going to be the aldosterones. So aldosterones act on the function of the kidney, ATP production, blood pressure regulation, again through the kidneys, regulating the plasma concentration. If we're thinking of the function of the kidneys, also affects the urination. So really what you would expect that happens if we have a hormone that affects the kidney. The reason why we need to know that there is a hormone known as aldosterone is that there is a condition called aldosteronism. That's when there's too much aldosterone being produced. And all of these processes that we saw here would be like to a power of 10. They would be amplified. They would be more faster or more efficient than they should be. So that can cause issues if the kidneys function too fast, if there's too much ATP done, if blood pressure regulation is to the extra scale, plasma concentration, urination is again amped up. So that's the aldosterone. The other hormone from the cortex of the adrenal gland that I want us to look at is going to be the glucocorticoids. And this is a group of hormones, including cortisols, hydrocortisones, corticosterones. If any of you, for example, have severe allergies or issues with skin, you might be given topical cortisone that you apply. Also in certain cancers, for example, we might inject cortisone to a patient. In certain damages, injuries, we might use cortisone injections. So many times, many purposes that we can use these glucocorticoids. Mainly, they act on stress responses. So if you think, why would we inject someone who has an injury, say, for example, a knee injury? Why would we inject cortisone there? Well, that helps with recovery by being the hormone that would be even normally secreted in response to a stress. So when you're going through a period of stress, you are able to overcome some of your normal limits because your body compensates for that. And we see that often, for example, after exam week, midterm week, finals week, students have been running on stress hormones. So once they are through that really demanding time, many of them get sick. So that's because you've been simply going on with your body producing stress hormones so you can get by. So there's all these actions that we can find that relate to the glucocorticoids affecting the metabolism, stress responses, glucose regulation, and so on. I want to highlight the stress responses for our purposes today. In terms of the hormones from the medulla of the adrenal gland, there's really two ones that I want to highlight, epinephrine and norepinephrine. What these two do, again, you can think of responses at the time of stress. So these help with bringing up the heart rate, reorganizing the blood distribution in the body, and so forth. When we might see this? Well, if you think back to when we talked about the autonomic nervous system in BIO201, there was two branches to the autonomic nervous system. We had sympathetic nervous system and parasympathetic nervous system. Well, sympathetic nervous system was all about fight or flight responses. So you were either going to run away from the danger or you were going to fight for your living. Well, these hormones, epinephrine and norepinephrine, act on this branch. The other branch, which was the parasympathetic nervous system, that was when you were simply resting and digesting. So the difference would be that at the sympathetic nervous system branch, we are directing blood to the muscles, to our sensory organs, our heart is racing, blood pressure races. We don't really put resources on digestion or urine production, things like that. It doesn't matter if you digested your meal, if you're going to die. You're focusing your resources on those physical responses that you can get away from the danger or you can fight for your living. Whereas the parasympathetic branch was all about now there's no risk. We've had our Thanksgiving meal. Let's focus on digesting that. So in that case, on parasympathetic nervous system branch, the resources would be directed on digestive system and urinary system, processing those energy that we've taken in and getting that to the use of the body. So kind of this next point that I've listed here as the hormone, the adrenaline, that relates to what we were discussing just up there. So I'm going to add that as the third hormone relating to what we were discussing there. So just kind of reminding you of that sympathetic branch of the autonomic nervous system. The next that I have here, and the order you might notice is just simply based on the alphabet because I got lazy. I didn't want to say that one is more important than another. Next that I have listed here is going to be our primary sex organs. So for the females, that's going to be, of course, the ovaries. Let's take this color. So what ovaries do produce the egg cell. That's an important function of the ovaries. But from the point of view of our structure, what we are more interested on is the hormones that the ovaries produce. And these two hormones are going to be the estrogen and progesterone. And we will talk about those as well when we look at the female reproductive system in a little more detail later on this course. But at this point, I do want you to be just aware that the two main hormones for the female gonads, so for the ovaries, are going to be the estrogen and progesterone. Let's have a look of those for the male primary sex organ. So that's going to be, of course, our testes. And testes, what they do, they produce, of course, the sperm, so also known as the spermatozoa, which is the male contribution to the reproduction. Well, that's not the only thing that the testes do. The hormonal function of testes is the production of the testosterone. So again, be aware of that, and we will discuss in more detail about that when we look at the male reproductive system. Next one, we actually get back to the kidneys that we saw already earlier on here. So don't let that throw you off. You can just make a reference here. We remember that they were these bean-shaped structures, and we'll look at them in greater detail later on. But in terms of the kidneys, the hormone that I want you to know for these is gonna be renin. So renin is a hormone that we look, is a hormone that plays significant role, for example, in the control of the blood pressure. And while we are talking about blood pressure later on this course, you will see, for example, renin-angiotensin mechanism, which is responsible for the blood pressure regulation. So that's an important hormone to remember, produced by the kidneys. You might ask, where does this word come from? Well, this word actually comes from the fact that whenever we speak about the medical field of kidneys, we don't say that you're going to go and see a kidney doctor. We go and say that you're going to go and see a renal doctor. Same as you don't go and get images of your kidneys through ultrasound or MRI or CT. You actually go and get renal imaging done. So that's just the proper scientific term for that, and that leads us to the name of the hormone, which is going to be the renin. So remember, main function of the renin, all about to do with the blood pressure control. The next one that I have, I'm going to come back to leptin in a second. I want to talk first about the pineal gland. And if you had a chance to look at the video about the dissection of the sheep brain that I added to the first module that we had on the week 2 module, I actually showed there the pineal gland. I'm not going to ask a single question from that video. That's just simply because someone was interested about this topic. And I was like, well, since we can't dissect now in a virtual setting, I might as well upload a video that I've done before and show there. So pineal gland is this kind of a small pea-shaped gland within the brain, deep in there. And what it does, it releases an important hormone to us. And the hormone that is released from this pineal gland is going to be melatonin. Notice it's not melanin, it's melatonin. So what melatonin does, it regulates the sleep and wake cycle of ours. So for example, individuals who have, blank under leptin. Oh, we'll come back. Yeah, we'll come back to the leptin in a second. I'm gonna address that just simply because we'll go slower as well, simply because leptin is something that I want to speak separate. So let's focus on the pineal gland. Pineal gland, like I said, located at the within the brain, inside by the third ventricle. Main function, production of melatonin, which regulated the sleep-wake cycle. Where we see the melatonin being used, especially as a supplement, is individuals who travel between multiple time zones back and forth. So they might need melatonin supplements. Same for the individuals who have trouble falling asleep or staying asleep. So they might be given melatonin. Again, a drug, again, a hormone that then acts as if it was produced by pineal gland. Times when we give this might be when pineal gland would not normally be active. For example, if you are traveling between different time zones, we do have an internal circadian rhythm, so we do internally go asleep when we should be going asleep. But if you mix up that internal clock, you might need to help that by adding this artificial hormone when our own body would not be producing that. And like I said, I'll come back to the leptin in a second, but I want to mention pancreas since we saw that already earlier on. With the pancreas, what was noteworthy was, sorry, I lost my tools, was that we love whenever there's something that's a little odd or unusual. And in this case, pancreas was the gland that was both endo and exocrine gland. It's going to be located underneath the stomach. We'll talk more about the digestive system later on this course. Right wrapped up to the arm or within the first part of the small intestines, which is known as duodenum. You don't need to worry about the name. But you'll notice that it's kind of this carrot-shaped structure, a little bit thicker than a carrot from the right end. And what pancreas does for the endocrine system was that it regulated the blood sugar levels through two hormones. These hormones are going to be insulin and glucagon. So for the pancreas, remember the blood sugar regulation through the hormones insulin and glucagon. You might have heard about someone or might know someone who is diabetic. So people who have a diabetes, there's a few different reasons. You might have either type 1 or type 2 diabetes. Type 1 diabetes is the one that you're born with. So in this case, your body doesn't either produce or respond to or it doesn't produce enough insulin. And in this case, these individuals often end up having to take supplementary insulin injections for all of their life. So that's something that you're normally born with. Type 2 diabetes is late onset, so that comes usually only later in your life and can be managed fairly well with lifestyle changes. So if you do some positive, healthier changes, you might not have to rely on any pharmacologics if you end up diagnosed with type 2 diabetes. But those were two hormones to do with the blood sugar regulation. And I'm going to talk about parathyroid gland again once I first talked about the thyroid gland, because I just want to be sure that you don't wonder about the relationship between those two. So we'll come back to the parathyroid and thymus. Let's focus on the thyroid gland. Where do you find a thyroid gland, what it is, and why do you need to know about it? Those would be good topics for us. So thyroid gland is located at your neck, just below the larynx, and sometimes that's described when I'm describing it or I'm reading descriptions of it as being shaped like a butterfly. To be honest, I think it is a bit of a stretch in the description. We do have two lobes that are connected by this short part. So that's, I guess, where the butterfly shape comes from, if you wish to see there a butterfly. But what thyroid gland does, it releases a hormone known as T3. And T3 gets changed into T4 hormone. And what these hormones do, they affect our metabolism. So often when we have patients who have weight management issues, we do look that could it be because of the thyroid hormone issues? Most often it is not, but sometimes it is. So you can have over or underactive thyroid gland. Let's start with the overactive thyroid gland. These individuals who would produce too much T3 would be able to eat anything at all, and they still cannot maintain the weight. So they are suffering almost from malnutrition, not getting enough of the resources from the food because their thyroid is overacting. It's overly active. So how we can treat that? We can use certain pharmacological interventions where we either slow down or bind some of these extra T3 hormones. Another thing that gets done sometimes is that part of the thyroid gets removed just physically. So that's a little bit easier if you have too much something. Let's look at the situation where you have too little thyroid hormone. So your thyroid gland does not produce enough T3, and it doesn't then as a conversion of that, we don't get enough T4. Well, these individuals, no matter how little they eat, how active they are, they just seem to put on more and more and more weight. So that would be a problem as well, because it's nothing to do with what they eat or how their lifestyle is. It's everything to do with not enough thyroid being produced and secreted. So I think that is all you need to know about thyroid gland. Remember, all to do with the metabolism. That's the important take-home message there. There are some clinical cases, Graves' disease, having an edema behind the eye, and so on and so on. If you're interested about those, you're going to love when you get to the pathophysiology courses. You're going to enjoy that. But on this level, we're not really going to dive into that. So now that we've had a look of the thyroid gland, I'm going to go backwards and have a look of these parathyroid glands. And the reason why I wanted to talk about thyroid gland first is because these parathyroid glands are located within the thyroid. So these are tiny yellowish or brownish dots, if you wish, within the thyroid. So I'm just going to add them to our diagram here. So there are these dots that we see here within the thyroid gland. And what they do, they produce a hormone called parathyroid hormone. And why parathyroid hormone is important, you might be thinking that, well, that's just giving another name for the parathyroid glands, and what they produce is the fact that parathyroid hormones are involved with the homeostasis of calcium. And so far, we have not talked a lot about calcium, but calcium is super important. Ooh, I noticed that I drew over our term there, so you can see everything. It's super important for multiple processes in the body. So, for example, in the function of kidneys, urine, intestines, digestion, skeleton, vitamin D activation. There's a lot of places where we keep seeing when we look at the physiology of various processes that calcium is involved. So that's something where we need to have parathyroid glands present, and they need to be working correctly so that all of these other processes in the body can work. So just put it at the back of your mind again that it is something that is an important hormone to do with calcium, and you'll see all these examples of calcium later on as we go through the material. Next one that I want to mention is gonna be our thymus. So why thymus is important is again kind of just prepping us for something that we will see later on. It's all to do with your immune system and immunity. So we will talk about the immune system later on as a separate chapter on its own. But at this point, I want you to know that it is the T cells that go to mature within the thymus. And these T cells are important cells when we are going to be looking at the immune system. What makes thymus an interesting one in terms of the anatomy is that we can see thymus within kind of the lower neck on top, on the chest region, right above the heart, roughly. It's going to be very visible and very obvious structure to notice in newborns, in children, and in young people. But then it starts shrinking away and gets replaced by the adipose tissue, so by the fat tissue, and also by the connective tissue. So in older people, in adults and elderly population, it's really hard to actually find a thymus within their body. So it's kind of a tricky one to deal with. So it shrinks over the age. But all about thymus and T cells, again, we will get back to it later on. I think that now it's going to be a good time for us to go back to, I promise that we will come back to leptin. And I'll tell why I left it a little later. Well, leptin is one of these hormones. So remember, leptin is a hormone. It's one of these hormones that it's hard to pinpoint to any particular body part. Instead, it's easy to pinpoint it to a particular tissue type. So we believe and know at this point that it is the fat tissue, so adipose tissue, that produces leptin. And leptin is a hormone that controls your appetite. So if you don't have enough leptin, or if you have too much leptin, we will have problems. So for example, what we've done in lab experiments, and I was lucky enough to be part of a group that studied this, we took mice, lab mice, and removed their leptin receptors artificially. Or we might have removed, so what we did, we removed their responsiveness to leptin. When they do not have any receptors for leptin, they will just keep eating and eating and eating. They have no control over their appetite. And they will eat to the point where they get sick, still keep eating, and eventually die as a consequence of overeating. And I'm not just talking that, oh, they got a little bit choky and watched too much TV and died because of that. No, they will actually eat themselves until their organs can't handle it. Well, you might wonder, is this only in the lab animals? No, we do sometimes, and this is not too common, but it sometimes happens. We do get, especially younger patients that are just putting weight without any control, and we can't really find a reason for that. They have appetite that cannot be satisfied in any way. Well, there's been some very successful results by giving them artificial leptin. And all of a sudden that has brought like a control to their appetite, that they don't feel hungry all the time. They actually are able to manage the amount of food that they eat. They feel satisfied at some point, and they have been able to return to the normal body weight, normal body size. So that's just another hormone that I want you to be aware of, but you don't need to know in detail of how it works or what happens in a minute detail as such. So I think that covers a review of the hormones. I'm just going to check, have I skipped over anything, but I think we covered everything. I know that was a lot at once, but that's probably as intense as this session will get. And what we have left, and I know that we didn't cover it during our review sessions of the endocrine system, we have some basic theory about endocrine system to look at. And this basic theory, although we didn't cover it on the review session, it is still something that I want you to know for the exam. So we're going to go through it. We're going to go through it kind of on a more vague scale than digging into minute details, but it is something that I want you to know. So I think when we're talking about hormonal system, endocrine system, it would be a good idea for us to be able to define what is a hormone. Well, hormones are chemicals that act throughout the body and can act through long distances. So they're not just local, as we saw with the exocrine glands. Endocrine glands, endocrine system acts throughout the entire body. So that's a very nice definition for the hormones in case you're kind of wanting to have one. And there's two types of hormones, two types of chemicals that I want to highlight here. And we're going to start with the autocrines. So autocrines are these chemicals, or hormones if you wish, that are secreted from the cell, and they might be secreted either outside the cell or within just the same cell. But these hormones act on this very same cell that secreted them. So what I'm drawing here is a nucleus inside the cell and cell wall. So autocrines act on the same cell. Whereas paracrines, they instead, again, don't travel a long distance. So they don't travel hugely far, but they travel to cells that are nearby, but not the exact same cell that produced this particular chemical. So those are known as paracrines. In terms of the chemical structure of the hormones, I think that I just kind of wanted to include it for the sake of completedness. We have some amino acid-based hormones. Hormones are often synthesized from cholesterol. That's how we get our steroids. So your body does need to have some amount of cholesterol, but all the cholesterol that you need is actually produced by the body itself. You don't need to get cholesterol from the food. So don't go crazy. Don't eat that cholesterol-rich food just because you think that you need it for hormones. And then there's this third group. We're still kind of debating, are eosinocytes hormones or are they paracrines? So this classification as autocrines and paracrines sometimes gets a little muddy. Are they hormones or are they just chemicals? Well, are all chemicals hormones or not? So that's something that I don't think we actually have a definite answer, but I would like you to be able to separate the autocrines and paracrines from each other. What I want to talk about next is a question that's quite a fundamental one, and I think that it comes up whenever I discuss with students, that if the hormones travel everywhere in your body, why don't we see them acting in every part of the body then? That would make sense. But there is a very good explanation for that. For a hormone, even though it travels everywhere, for a hormone to have an effect on a cell, that cell must have a receptor for that particular hormone. So you might have cells that are exposed to many different kinds of hormones, but unless there is a receptor for that particular hormone, it will not trigger anything to happen. So we noticed that the hormones and the receptors are like this key and lock mechanism, that they must be an exact fit. Roughly similar one will not work. It needs to be a perfect fit. So what these receptors are, they are located on the surface of the cell, on the cell membrane. And this hormone travels and binds with that receptor. And only if it's a correct fit, then that receptor will trigger a series of changes inside the cell. And we'll have a look at this concept in a little bit more detail, but this is kind of the large picture. So if it's a fit, then the target cell's activities change somehow. It might be that we activate certain genes within the DNA, or start producing certain proteins, or so on and so on. And this brings us to talk about that not all hormones are alike. Some hormones are water soluble, whereas others are lipid soluble. And that reminds us about the cell membrane of the animal cells. So phospholipid bilayer. You remember that the outermost layer of the cells is made of lipids. Well, if your hormone is water soluble, there's no way that it can cross this lipid membrane. Instead, what needs to happen, that on this lipid plasma membrane, it needs to act on a receptor that's located on this membrane. And then this receptor triggers a series of events inside the cell. So these hormones that are water soluble require these second messengers inside the cell. Lipid soluble hormones instead, they are able to cross inside the cell through this plasma membrane, and there they trigger whatever changes need to happen, such as activating genes or changing how the cell functions. I've also included here a little table that I think that I prepared for your PowerPoint presentation back then about the differences and similarities, comparing and contrasting lipid and water-soluble hormones. It's there for your use if you want it, but I don't think that I really asked a lot about that. Now we get to one of the basic concepts that I already mentioned earlier, and this is going to be such a vital concept that even though it feels like you already know it, I'm going to take the time today to go through it, because later on this course, I will simply keep referring back to this concept with the assumption that you're familiar with that. And a lot of processes that we see in the body, we will explain through this. So homeostasis referred to the keeping the internal environment, the conditions inside the body, as consistent. So it's the consistent, no changes that we're after when we're looking at homeostasis. And you can find all these beautiful diagrams where we have seesaws, we have arrows going here and there, and they get very complicated very quickly. I have condensed everything you need to know about homeostasis into 4 or 5 steps, depending on how you approach it. So what our body wants to do, our body detects whenever there is a change of some kind. This change is detected as a stimulus. So something is not remaining as a consistent, as something that we are used to being a consistent condition. So the example that I think that I used with you when we were discussing about homeostasis was, for example, body temperature. So on this line, you can imagine it as a scale of different temperatures. We have very hot temperatures up here and very cold temperatures down here. And your body needs to be kept within a certain range of temperatures. And if it changes from that range, then there's gonna be a need for the homeostatic control mechanism loop to kick in. So let's have a look of that. How this change is detected, it's gonna be a sensor or a receptor of some kind. And this receptor or a sensor itself doesn't really give to what it notices a meaning. Instead, it passes a message to the control center, which is typically, in case of animals, in case of humans, the brain. And within this control center, we have a set point. And set points is an range of acceptable values. So for example, for the human body temperature, we keep human body temperature around 37 Celsius degrees at all times. If it changes much colder, let's say to 36 or 35 Celsius degrees, that will be a problem. If it gets much colder than that, then you will probably die. Your body system starts slowing down, your processes won't work as well. If it gets much higher, let's say to 39 to 40 Celsius degrees, your enzymes will slowly start to deteriorate, to degenerate. And again, if you don't have enzymes, your proteins are affected. And again, if it gets much higher, you will die. So we're keeping this temperature within this set point, within this range of values that we see here. Well, if we deviate, if the stimulus is off from this range of acceptable values, effector will kick in. And what the effector will do, it can be a physiological response or it can be a behavior that we do, but it will always aim to stop this change from happening. So what you do in electric diagrams or engineering diagrams, when you're stopping something from happening, it's going to be this line with a line on top of it that stops from happening. Stimulus now no longer would happen, and we would return back to that normal set point. So in terms of the body temperature, it could be, for example, that you start to shiver, you start to produce more heat, you put on more clothes, you curl up, you move to a warmer environment. So that's what normally happens. And what we see here is an example of negative feedback. Negative feedback stops this change from happening. And that was the most common kind of response in a homeostatic feedback mechanism loop. Well, there was an exception to this, or a couple of exceptions that we've already mentioned, and these were positive feedback. In case of a positive feedback, stimulus gets detected and processed within the control center, and we will actually promote that stimulus to happen even further. And the example of that was the childbirth. We saw when the child is coming out, that only promotes the delivery even further. Same goes for the scab formation. When we start bleeding, the clot forms, and a little clot forming causes more clot formation to take place. We saw that also with lactation, the suckling of the nipple promoted for more milk to be released, which makes the baby suckle that nipple even more, which again releases more milk. So this is a positive feedback. I think there is a fourth example, and I just can't think of what it could be. But for our purposes, these are the three that you need to know. So I already mentioned this part earlier on our review, that when we look at the nervous system and endocrine system, that often there is overlap between the two. So hormones can trigger for the nervous system to kick in, and nervous system can trigger for the hormonal system to kick in. So there is overlap and interaction between the both. What else should we know? We should know that how efficient a hormone is can depend on three different factors. So it can depend on how much of that hormone is in the blood. It also depends on the amount of receptors in the target cells. So if we don't have any hormone, it's not gonna work very well. Or if we have lots of hormone, but there's no receptors for it, it's not going to work very well. And also how tightly this hormone and receptor will connect. You remember it needed to be a key and lock mechanism, so perfect fit. But how tight that fit is, so that will also have an effect. So all of these three factors play a role on the effect of the hormones. Well, sometimes one or two of these is working really well, but there is an issue with the third factor. And let's have a look at that. So we can have situations where we have very little hormone present. So like here, if we have very little hormone present, body still will want to notice when that hormone is released. So in response to that, we actually form more receptors. This is known as upregulation. So low hormone levels cause more receptors to be produced. This is known as upregulation. In certain cases, we have so much hormone there that our body and the receptors are constantly going like, yeah, there's that hormone. Well, how body responds to that? If there's a lot of hormone present, let's remove some of those receptors. So let's reduce the amount of those receptors so we don't get that high of a signal all the time. This is known as down regulation. The final thing I want to mention, a couple of things here. Hormones can be either free in the blood or they can be bound into carriers or to plasma or elements of other kind that are not blood on its own. And we of course have to either inactivate or remove some of the hormones once they have done their job. And this is normally done by kidneys, liver, and certain enzymes. A concept that I wanted to introduce here is a half-life. Half-life refers to the time required for the hormone levels to decrease into half of the initial hormone level. So if you think that we're either inactivating or removing hormone, here is a time on the x-axis, and on the y-axis we have concentration of a hormone. So let's say that initially we have high amount of the hormone, but once it has done its job, the amount of it starts to decrease. Well, this point where only half of its initial amount is present is gonna be our half-life. So we can say that the half-life of this hormone happens within this amount of time. So that's simply what that term refers to. But that's an important term to be aware of. One last concept in terms of the hormones and endocrine system is that hormones don't act alone. One hormone can have an effect onto another hormone. And there's 3 different terms that we need to know here. And these terms, I would be able to recognize these. So I would encourage you that you're able to match the term with the description. That would be a good thing for the exam. So let's have a look at these 3 different kinds of definitions. And we're going to start with the permissiveness. So in this case, we have this hormone that is supposed to do something. But it needs another hormone to be present so that this initial hormone can do its job. It can have the effect that is needed. So this is permissiveness. It's almost like giving a permission to this hormone to do its job. So this second hormone is simply allowing this initial hormone to do its job. The second term that we have is going to be synergism. So in this case, we have this initial hormone, and the presence of the second hormone actually makes this initial hormone to act even more. This initial hormone would have anyways have an effect, but when you combine it with this second hormone, it's going to have much larger of an effect. And the final situation that we can have is antagonism. You might have heard that in most stories or most films, most books, there's an antagonist. It's something that's the villain of the story. Well, in this case, we have the initial hormone, and then we have the second hormone, which is an antagonist hormone. It stops this initial hormone from doing its job. So that's an antagonist hormone. So at this point, we have covered everything you need to know about the endocrine system. I know it took longer than I had anticipated. There was a lot of detail. So I will still stay around and go through a chapter on blood. It's going to be shorter, but I totally understand that you might have places where you need to be. I'm expecting to spend about half an hour on that at the maximum. But I understand if you have to go, there is going to be, I'm recording this session. It's going to be made available, I hope tonight, overnight, to upload it on the YouTube. So I appreciate if you stay, but I will not be offended if you have to go. But at this point, if you have any questions about the chapter on hormones, just let me know. I know it's a lot of information, but you can have these notes with you in the exam. So what I'm going to do, if I don't hear anything else, we're going to move on to have a look of the blood. And like I said, this is going to be a little bit quicker to look through. So let's have a look everything that we need to know about the blood. So again, just to put this chapter into a greater concept that or context, that what are we going to be talking about on this course? This chapter is going to be all about the blood, which is really the transport media within the cardiovascular system. But we are going to be looking after this chapter also the heart, which is the pump that moves this transport media, and then we will be looking at the tubing, which is kind of the which is the blood vessels where this transport media will travel. So just so you know, there is going to be more about the cardiovascular system later on, but we are only focusing on this one, on this exam. What is blood then? That's a good question, and that's one question that often catches students by surprise. So you remember we had 4 main tissue types. We had our connective tissue, which actually normally was things like bones, ligaments, tendons. And connective tissue connected 2 parts to each other. But we will end up noticing that blood is a connective tissue as well. Although it doesn't physically form this tight connection that we're used to seeing, what instead happens is that it connects different body parts to another by transporting materials. So keep that in mind there. And really, if you want to give a description, what does blood do? Blood is part of our internal transport system in the body. Where is blood made then? Well, blood is made within the bone marrow. And here is the first time that I'm going to mention these 2 terms, or especially one of these terms. And these are going to be important terms for us to be comfortable with. So when we talk about production of the blood, and this means all of the blood, not specifying one particular component of the blood, we talk about hematopoiesis. So hemato term refers to the blood, and poiesis refers to the production. So when we're discussing in general production of the blood, we talk about hematopoiesis. We will see later on that we can talk about production of different components that make up the blood. And in these cases, we just change the first part of this term. We talk about something else and poiesis. So just something to be aware of, that's going to be an important one. Kind of also setting our scene, there's about 5 liters of blood in a human body. And reason why we need to go through this chapter about blood and why blood is so important if you're heading to a field of healthcare, nursing, you're thinking of medicine, dentistry, various different fields where you can go, is that drawing blood from a patient and then analyzing what's in that blood is probably the most common test that we do in clinical work. And I read some time ago a study that I'm pretty convinced was fairly accurate, that about 70% of the decisions that a medical doctor does is made of simply looking at the results of this analysis of the blood. So really, this is an important way for clinical decision making. Okay, let's have a look at blood in terms of what is the function of the blood. And I've given you quite a few different ones, but there is one that I'm especially going to highlight. So blood is really a transport media. So blood is used to transport gases such as oxygen from outside world to the tissues that might need oxygen. It is used also to remove the carbon dioxide that is produced as a byproduct by those tissues while they do their job, and then it gets taken to the outside. Also nutrients, energy is transported within the blood. Any waste products from the tissues is transported within the blood. We just looked at the hormones. So a lot of things are transported within the blood. Other things that blood plays role is also regulating, for example, fluid balance in different parts of the body. Well, a lot of the blood is actually fluid, liquid, so it makes sense that that's an easy way to move fluids from one part of the body to another. Water is excellent in its capacity of binding heat, and since much of the blood is actually water, it's also a great way to move heat from one body part to another. Same goes for the pH regulation. Protective function of the blood. We will be talking about the immune system in a greater detail after we have covered the cardiovascular system. Also, if you cut yourself, you don't bleed to death. That's the protective function of the blood. You will form a scab. So really, those are the three groups that you can kind of find for the functions of the blood. But I would emphasize especially this transport role of the blood, if you want to have one as a major one of those. Well, at this point, what we're going to do, we're going to go through different parts that make up blood. And that's really all there is left for us to look at from the blood. So what I'm going to do to have some sort of a reference that we can come back as we're looking at different parts of the blood, I'm trying to draw here a picture of a test tube. It might not be the best picture, but it's the best I can produce now. So if you're wondering, what is that? That's a test tube. And within this test tube, we have blood up to this slide. So all of this is blood. And while we're reviewing this chapter of the blood, we will look at what components is this blood made of. And this actually is a good way of looking at this, what components the blood is made of, because this is exactly what we do when we take the blood sample of a patient and put it to a centrifuge and spin it. So as a result of this spinning process, all of these different parts that make up the blood have a different weight, so they travel to different parts of this test tube. So we're able to separate different elements of the blood by this method. So that gives us a good clue of what and how many and how much we should find from the blood. So let's start with the most common element of the blood. I think that that's always a good way to start. What do we find most? And most of the blood, about 55% of the volume of the blood, is going to be plasma. So you might have seen plasma before. If you haven't, it's kind of this yellowish or a straw-colored liquid. It's a little bit stickier, a little bit thicker than water in its consistency. But when we separate the blood into its components in a test tube by spinning it, plasma is the component that's going to be right at the top of this test tube. So about 55% of the contents of this test tube is going to be plasma. Plasma contains a lot of things. Mainly proteins are the ones that I want you to be aware of. And I mentioned 3 proteins, albumin, globulins, and fibrinogens as examples that are important. But in addition, plasma can contain a lot of other things, nutrients, gases, hormones, wastes, inorganic ions, and so on. So that's what plasma is. Plasma is really simply a liquid where everything else is. Because what we find when we study blood, that this is the only liquid part. Everything else in the blood is actually cells or parts of cells. So there's something that's tangible. We can really pick out one piece by piece of these other elements that make up the blood. Well, these are known as formed elements, all these other stuff than the plasma. So only plasma is liquid and everything else is formed elements, so this remaining part. And we're going to go through all of these formed elements that we need to know that the blood is made of. And these formed elements then in a blood that hasn't been centrifuged, they are mixed, floating around everywhere within this plasma. So plasma is like liquid, and then salt crystals would be the formed elements. But salt does actually dissolve the water. But something that doesn't dissolve, let's say sand that we put into the water. It might look like it's dissolved when we mix it a lot. It looks like it's all there, but over time it separates to the bottom of the sample. So let's go through remaining parts of what blood is made. Well, one part of the blood that I'm sure you've heard before that blood is made of is gonna be our red blood cells. And because there's a lot going on in the world and no one has unending time, often you'll notice that red blood cells get abbreviated as RBC. So that's just an abbreviation for the red blood cells. But really, it's not red blood cells that's the scientific name for this. The scientific name for red blood cells is erythrocytes. So that would be something to know. So erythrocytes is the scientific name for the red blood cells. And erythrocytes make up about 45% of the total volume of the blood. So we can add the red blood cells on this diagram that we had here. So almost rest that's in this test tube is going to be red blood cells. And red blood cells are the heaviest out of all the things that we see here. So they're going to be at the bottom of this test tube of blood sample that has been separated into its different components. So that's supposed to be our red blood cells there. That looks better. So let's have a look at what do red blood cells look like. Well, red blood cells look like these flattened discs that have this depression in the middle. So they're often described as being biconcave discs. They look like donuts that don't have the hole fully punctured through, or they look like balloons that someone has squashed from the middle, so it's round but not fully pinched through. If we were to take a cross-section of this red blood cell, I've drawn it like looking from outside, let's think that we're going to cut it into half here, down this plane. So we're going to cut it into two. What we end up seeing is that a red blood cell would look roughly like this. On the sides, it's thicker, and right on the middle, it gets thinner. So this is a cross-section of a red blood cell. And this shape is important for the function of the red blood cell, which we will talk a little bit in a bit. So the term how we describe this cross-section of the red blood cell often is that we say that it is a biconcave disc. So meaning that on both sides it's kind of squashed, it's concaving, it's caving inside, if you wish. So that's the description that we use for the red blood cells. Another thing that's probably quite interesting about the red blood cells is that you notice we call them anucleate. That means that they don't have a nucleus. But that doesn't really add up with what I've told you before, because I've said to you before that all cells have a nucleus. Well, red blood cells did have a nucleus back in the day during their maturation process, but then that nucleus gets removed or is removed from the red blood cell. In addition, all of the cell organelles are also removed from the red blood cell. So there's no cell organelles in this cell either. And when you think about the function of the red blood cell, which is simply to transport oxygen, that makes sense. If we had a bunch of cell organelles there, they would probably end up needing oxygen too, and they would use all that oxygen before red blood cell is able to transport what it is to transport to where it needs to transport it. So it makes sense for it not to have a nucleus and not to have any other cell organelles either. Well, before I get any further, I want to go back to here where I have written erythropoiesis, because this is an important concept that I kind of referred to earlier on, and I will ask some questions about these terms. So I said earlier on, if we look back, and I will come back to this view, I said that when we talk about production of the blood, we talk about hematopoiesis. So hemato was blood in general, and poiesis was making. Well, when we talk about production of the red blood cells, now we're going to talk about erythropoiesis. And you'll notice this word erythro refers to erythrocyte, which was the scientific name of the red blood cells. So the production of the red blood cells is going to be erythropoiesis. Well, let's have a look at how the red blood cell does its job of transporting oxygen. Well, what we find in the red blood cell, and I'm going to draw here just an imaginary red blood cell, really quick diagram. It's a blob. We end up finding within the red blood cell hemoglobin. And there's actually 4 hemoglobin subunits for each red blood cell. So they're located first, second, third, and fourth. And each of these subunits of the red blood cell is capable of transporting 1 oxygen molecule. So each of them is able to carry 1 oxygen with it. Well, if all of these 4 hemoglobin subunits have an oxygen bound into it, which is the maximum amount of oxygen that can bind into a single red blood cell, that is known often in diagrams as being bright red in color. So these cells, if you see diagrams where we're discussing, for example, about flow of blood, these arrows would often be bright red, or these cells would be drawn as being bright red. That tells us that there's a lot of oxygen in this blood. That's because these hemoglobin subunits are now described as being oxyhemoglobin. So they contain oxygen. So often these cells or this blood would be coming from the lungs. So it's being packed full of oxygen within the lungs. And now it's traveling to other parts of the body. Well, we also have an opposite of that. When blood has gone to the tissues and given away the oxygen that it was carrying, that was its job. Its job was to carry oxygen to the tissues. It now has very little oxygen left. And it's often described in these diagrams as being blue in color, when we don't really have blue blood, but we have this dark purplish blood. So this is typically coming from the tissues. And this kind of hemoglobin is described as being deoxyhemoglobin. So we will come back to this when we talk more about the transport of the blood and so on. But I do want you to be familiar with the difference between those two. Well, why does this all matter? Why do these 4 hemoglobin subunits within a single red blood cell matter? Well, it matters because each one of them could only carry 1 oxygen molecule. But there are other gases that are very eager to bind into those. One of those would be, for example, carbon monoxide. Where do we find carbon monoxide? We find when we have something that doesn't completely burn. So, for example, you have a fireplace. It's not fully out, but you close the fireplace anyways. And some of those gases that get still released by that final burning is going to be carbon monoxide. So you can get a carbon monoxide poisoning. You also have small amount of carbon monoxide in the cigarette smoke. So what happens? Carbon monoxide gets to these hemoglobin subunits quicker, and it binds into this hemoglobin and blocks it from picking up oxygen. So the place of an oxygen has already been taken up by carbon monoxide. This would be an example of why it is a bad idea to smoke. So you're not able to transport as much oxygen in your red blood cells. Okay, I think that we have to look just a little bit more about the red blood cells. This is gonna go rather quickly. So what we want with the red blood cells typically is that we want the amount of red blood cells that we produce is equal to the amount of red blood cells that we remove. This is because red blood cells live only about 100 to 120 days. I say only, but that's actually a rather long time for as a component of the blood to survive. Once the red blood cell is no longer good, once it needs to be removed, it goes to then to be broken into different parts. It gets broken into all the elements that make up the red blood cell. So we collect the iron back, we collect the heme group, and we collect the globin back as well. And we can use these again, recycle to make more red blood cells. But let's have a look of a situation where this production and destruction of the red blood cells is not in balance. If we have too little red blood cells, we talk about this being a condition where one has hypoxia. And if there's too much red blood cells, the blood becomes very high in viscosity. How do we control then the production of the red blood cells? Well, there's going to be a hormone that's going to be important here. And this hormone is often abbreviated as EPO. EPO stands for erythropoietin. So just because we were talking about hormones earlier on, I want to highlight this. So what happens is if your blood doesn't carry a lot of oxygen, your body will respond to that by releasing erythropoietin. And what erythropoietin does, then it makes more red blood cells. So it brings up the production of the red blood cells. Well, you can think of this as a homeostatic feedback mechanism loop. So here we have low oxygen concentration in the blood. That's our stimulus. That's noticed by, you don't need to know this structure, chemoreceptors of the blood. They're the sensor that gets passed to the brain, which is our control center. We have there our set point. How much oxygen should you typically have in the blood? And if you don't have enough, we now start to produce EPO hormone. And that EPO will stop the oxygen level from going down. It will produce more red blood cells. So this is how you see this homeostatic loop work through a negative feedback. Well, when would this happen in reality? Let's think of a group of individuals who might be very interested about this. let's think of athletes. Athletes want to have a lot of oxygen coming into their body so that they're able to carry as much oxygen as possible to their muscles while they're exercising. And in theory, that could give them a chance to work a little bit harder than their competition, and by that way, winning. Well, one thing that they can do, they can take erythropoietin and inject it into themselves and produce more red blood cells so they're able to carry more oxygen. This would be cheating. This would be doping. Instead, what's allowed, you can go to a high altitude. In high altitude, we know there's little oxygen around. Well, if there's little oxygen around, your body is going to be like, oh, we need more red blood cells. And your body starts naturally producing EPO, which increases the amount of red blood cells. So now when this athlete returns from high altitude training camp to normal altitude level, all of a sudden they have more red blood cells in their blood than their competition. So they're set for success. This would be allowed, and this is common practice before competitions. Other things that can affect the amount of the red blood cells in your body is, for example, iron from the diet and many other components of the diet. Are you getting enough carbs, lipids, amino acids, and so on? Finally, I want to discuss about the red blood cells, about a condition known as anemia. So when you have little oxygen in the blood, we're talking about you being anemic. Some of the signs and symptoms include that you appear very tired, pale. You might have breathing problems. You're constantly cold, chilly. There's few reasons why you might be anemic. You might be losing a lot of blood. So there can be, for example, internal bleeding. Or then you don't produce enough red blood cells. So production of the red blood cells is down. Or you destroy too much red blood cells. So red blood cell destruction is up. So there's those three reasons. It's usually one of those three that causes one to be anemic. And there's a special type of anemia that I wanted to discuss with you. And that special type of anemia was sickle cell anemia. And that was gonna be the topic of your extra credit question in the exam. So what I will ask in the exam is that write me a little bit about sickle cell anemia. It can be just a simple paragraph. I'm not looking for more than maybe 4 or 5 sentences. I'm asking you to tell me what is sickle cell anemia, what causes it, what consequences does it have that you have a sickle cell anemia, and how does that relate to malaria? And how does this all make you think about evolution? So let's go through answers to those. Remember that you can prepare this question ahead of the time by typing a little answer, one paragraph of this on a notepad or Word or somewhere else, and then just copy pasting it to your exam when you're taking the exam. So let's remind ourselves that normally red blood cell look like these biconcave discs. So it was this donut shape disc that was kind of squashed from the middle. Well, that's how it looks normally. However, if we have one error of a one base within the DNA, so you remember DNA was this double helix structure, and these lines were bases. If one of these is different than what it should be, that will have a significant effect on what kind of a red blood cell is produced. So let's have a look at that effect. So here we have now one base that differs from the earlier, and that causes when we are synthesizing a protein based on this DNA strand, that protein has one different amino acid from what we've seen before, and that causes the resulting protein and resulting red blood cell to take a totally different shape. These red blood cells look a little more like this. They're like crescent moon shaped in their shape. So this difference in shape of a sickled cells has a bunch of significant consequences. Well, the first consequence that we see is that they are able to carry less oxygen. So that's not great that they're not carrying as much oxygen as the normal red blood cells. The other problem that we are left with is that because of their shape not being this nice and smooth, they often also get stuck on where they shouldn't get stuck. So that would be an issue with the red blood cells that are sickled as well. So you might think that this really sucks for an individual who has a sickle cell anemia. That is true. They get less oxygen. If they are not super athletes or don't push themselves too hard, they still will be fine. But it's a little downer. Well, what if I told you that sickle cell anemia is most common within the sub-Saharan Africa and within individuals who come or whose family ancestry comes from the sub-Saharan Africa? So you might again think that, oh, that kind of sucks for those individuals. Well, let's remember what's going on in sub-Saharan Africa. One of the things that's common in sub-Saharan Africa is malaria. So what we end up finding that the area based on a map where sickle cell disease is common and area based on a map where malaria is common almost perfectly overlap. Well, why would this be important? Well, how malaria? Malaria is a parasite. It's not great to have a malaria, but malaria normally does. It enters inside the red blood cell, multiplies there in numbers, and once it's multiplied in enough numbers, it leaves the red blood cell by ripping and killing it in a process. So not great to get a malaria, right? Well, if you have a sickled red blood cell, turns out the malaria parasite is not able to enter into this type of a red blood cell. So it's almost like having a sickle cell anemia protects you from malaria, which was common in the area where you actually have a lot of sickle cell anemia individuals. So, and I don't tell you that you have to believe in evolution. It's up to you. But on this class in sciences, we do assume that evolution is something that happens. So we are discussing concepts considering that evolution is something that is present. So it seems almost because of this relationship between sickle cell anemia and malaria, like evolution has noticed a problem, which was the malaria, and it has fixed it by giving us a condition which is sickle cell anemia. So evolution has outsmarted something that would have been problematic for us. And we often need to think about before we start fixing people, treating people. Maybe there is a reason why we see things happening. Maybe certain diseases happen for a reason. I'm not trying to say that we shouldn't treat everyone equally, but we have to think back that, oh, could there be a reason why this is more common within this population than within this population? So that's just, I'm putting it out there. That is the question. That's the extra credit question in your exam. If you have any questions, I love it how you're finding that. That's a really good point. It is, you are lucky. I don't know if any one of you has ever taken preventive malaria medication. I have. And that's really hard on your body. These malaria medications that you take before you go to malaria areas, they, for example, cause many people, not everyone, but many people lose their hair as a result of those medications. So if you don't have to take it, you are kind of lucky. I agree. So if you write a little essay about that, don't go crazy. One paragraph is enough. You can get a bunch of extra credit towards your exam score. So I know that was a lot to take in. We're next going to look at the white blood cells, and this is very quick. And the final one that we're going to look at is going to be platelets, and that's like maybe 4 lines or so. So we're going to move on to have a look of the white blood cells. And white blood cells, again, often abbreviated as WBC. The scientific term that we use when we discuss about white blood cells is going to be leukocytes. So you see it there written. I just put it so it's jumping out on your vision. And the reason why I want to mention the word leukocyte is that when we discuss about the production of the white blood cells, I will come back to all that I jumped past. When we talk about production of the white blood cells, we talk about leukopoiesis. So notice how the term poiesis for making of remains, and we just change the term in front of, when we talk about white blood cells, it's going to be leukopoiesis. And again, I will come back to the rest of this. I just wanted to show that in terms of the term leukocyte. Well, leukocytes make about less than 1% of the blood volume. So not a lot in terms of the amount of them in there. And if we go back, I will come back to this. If we go back to this test tube that I drew, the white blood cells would often layer themselves here in between the plasma and in between the red blood cells. This area in between those two is known as Buffy coat. I'm not going to ask about it in an exam, but just so you know. So let's have a look why white blood cells are important. Although we have a small amount of them in the blood, if white blood cells are needed, the amount of them tending to grow. So white blood cells simply end up replicating, producing more and more and more cells if they are needed. This is when your immune system gets activated. So, for example, if any of you have already received or is thinking of receiving COVID vaccine, what it does, it activates your white blood cells. So we produce these protective white blood cells that will prevent you or protect you against the COVID. And during that time, the number of white blood cells will be a little bit higher than normally. And then those protective white blood cells will remain in your body and circulate there. And you have a protection. So what white blood cells really do, they do defend you against diseases. So that's their main function. And there's going to be 5 different types of white blood cells that I want you to know. In the exam, you will not need to be able to tell them apart one from another in terms of how they look. In the practical, I think that you are asked. So I think I gave you pretty good pictures in the lecture video. So I'm not going to go through on this revision session how to tell them apart in too much. But look at that lecture video, look at your practice anatomy and so on. But we will look at what are those 5 different types of white blood cells that we have. And we're going to start with neutrophils. Neutrophils are the most numerous of the all white blood cells that we have. So about 50 to 70% of all white blood cells are neutrophils. They are, in case you're thinking what they look like, and I refer you back to that lecture video, their nucleus is made of multiple lobes, it stains very dark, their cytoplasm contains granules, so it's like a dotty in appearance. What they do, they play a major role in immune defense responses. The second most of the white blood cells that we have is gonna be our lymphocytes. Somewhere around 25 to 45% of all the, I will come back to the ones that I'm jumping past, of all the white blood cells in the body are gonna be lymphocytes. So I'm just adding that here. Lymphocytes have a large spherical nucleus that takes almost entire cell space. The cytoplasm has no granules. So we describe that as being agranulocyte. The A means not granulocyte. What it does, lymphocytes play a role in antibodies and immune responses. So where they are produced or mature depends on whether they're produced or matured in thymus or bone marrow, and that gives the cell its cell type name, whether it's T cells or B cells. So that's our lymphocyte. The third most common of the white blood cell type is gonna be our monocytes. That's only about 3 to 8% of all the white blood cells are monocytes. I didn't mean to highlight all of that. Their nucleus is a little bit more like kidney shaped, and again there's no granules, so agranulocyte in the cytoplasm. And what they do, they're phagocytic cells, so they eat anything that shouldn't be in the body, they engulf it inside them, and then go inside them, they kill what they engulfed. So they remove non-wanted material. That's the monocytes. This fourth most common type of white blood cell is gonna be eosinophils. Eosinophils, about 2 to 4% of all the white blood cells. They do actually have two lobes of a nucleus, so it's called binucleate, and they do have those granules in the cytoplasm. What they do, they are involved especially in allergic reactions and act against parasites. And the least common of all the white blood cells is gonna be our basophils. They're only like less than 1% of all the white blood cells. So that's our basophils. They have, again, binucleate nucleus that stains dark. They do have granules in their cytoplasm, and they play a role also in allergies and inflammatory responses. For example, antihistamines help us to keep the basophil amounts in control. So if you have allergies, you might end up taking antihistamines. Allergies are simply your white blood cells having gone wild. So that's why you would take those to limit the production and amount of basophils. Well, you'll notice that I wrote the order of how common these different white blood cells were here. And the order is listed here as neutrophils, leukocytes, monocytes, eosinophils, and basophils. So I could very well ask in an exam that lists the white blood cells that I'm giving you here a list in the order of how common they are. Well, if you remember the next sentence, it's going to be easy for you to do. Remember that you should never let monkeys eat bananas. That's a good mnemonic. We love mnemonics in the field of anatomy and physiology just to make it easier to remember things. So if you remember that you should never let monkeys eat bananas, you're able to organize the white blood cells from the most common to the least common. Okay, we already discussed about the production of white blood cells, and the two clinical relevances that I'm going to mention regarding them are going to be our leukemia and leukopenia. In leukemia, you have too much white blood cells. Your body produces too much white blood cells, and often that's an indication of something more severe. In leukopenia, you would have too little white blood cells. That could be also problematic. So that completes everything that you need to know about white blood cells. And the very, very, very last thing that you need to know in preparation to the exam is gonna be our platelets. So platelets, I didn't mean to do it that color. Let's change the color. So let's have a look of the platelets. Platelets, just like the white blood cells, form only less than 1% of the entire volume of the blood. I will come back to this, but if we add it to our diagram that we saw here, it also is gonna be on this area known as Buffy-Coes. So between the plasma and red blood cells. So let's have a look of our platelets. So platelets are not actually full cells. They're just fragments of cells. And what their function is that they help us with clotting. So when you cut yourself, you will slowly start forming a clot there. The scab forms, and that prevents you from bleeding to death. So it's a really good thing to have platelets. Platelets only live for about 10 days in your body, so there's constantly new platelets being made. And I mentioned to you guys that, for example, I lack naturally platelets. I have to take artificial ones. So if I didn't take, I would just keep bleeding. So platelets are important ones. They prevent you from bleeding from death. So that's all that you need to know really about the blood. I did include a little bit about the blood typing. I have a video about that, and that video really covers everything. Also, this text is pretty good of explaining what's important when you do blood typing is that if you give blood transfusion to someone, the donor and recipient need to be a good match. If they're not a good match, you can actually end up killing that individual. So for that reason, we pay attention that how well do they match. If someone has a blood type A, they can receive blood, of course, from another person who's blood type A, but they can also receive blood from someone who's blood type O, because that individual is a good match with them. For blood type B, they can receive blood also from another B and also from a blood type O donor. If your blood type is AB, you can receive blood from any donor, whether it's A, B, AB, or O. If your blood type is O, you can only receive blood transfusion from another O donor. So the full story why this is and how this works is in the lecture videos, but what we just looked at helps us to know that type AB can receive blood from anyone. So for that reason, blood type AB is known as universal recipient. They can receive blood from anyone at all. Well, we also noticed that if you're blood type O, you can donate blood to anyone at all. Everyone can take your blood. So that's why blood type O is gonna be universal donor. So they can donate to anyone at all. And the final thing that we discussed that sometimes you hear that some blood types are positive or negative. That refers whether someone is positive or negative for Rh factor. And that has to do more of if the blood of the mother and the baby that's still developing is different in terms of their Rh blood type, and the bloods end up mixing during the delivery. And I've been in quite a few deliveries, and I've never seen a delivery where the bloods wouldn't end up mixing. Well, then the second pregnancy, after that pregnancy, can be problematic because the blood and antibodies in the mother's blood can end up causing issues against the blood type of the baby if it differs from that of the mother. And the blood of the mother can actually end up attacking that baby. And that can be very, very problematic. So Rh positivity or negativity was really an issue only in terms of if we had a second pregnancy. And if there had been mixing of the blood of different blood types, Rh positive and Rh negative, during the delivery of the first child. So that's all you need to know for this exam. I know that was a lot, and I know that I kept you for quite a while. So I'm going to jump now back to the PowerPoint, and we're going to wrap up. I'm really appreciative that you sticked around. Trust me, I always take those notes of those, and I keep it into account when I also look at the final grades at the end of the course. So last thing that I want to say about preparing for the exam is that if you feel that you would like to get help in terms of tutoring, you are a little lost, or even if you're not lost, one option that you have is to use our tutors from the college. Because we are not in face-to-face phase yet, because of the pandemic, you can contact our tutors through Cranium Cafe. That link is pretty much everywhere. You don't need to have an appointment. You just show up. If a tutor that you want to talk with is free, they will talk with you immediately. Then, if they're not free, you can request them, and once they're free, they will talk with you. The other thing that you can do if you feel that you need a little help, you want tutoring, you want a little bit of advice, you can always contact me. And I'm happy to set up aside a time for one-on-one virtual meeting, or we can even do groups, or we can do whole class, anything if you have questions. So I'm here for you to help as well. And that really covers everything I had in mind. If you have any questions, I'm so happy to answer those. If you don't have questions, I think we covered everything that we needed to do. I know that that was a lot in terms of the study guide, but remember, now that you filled in that study guide, every answer to every question in the exam is in that study guide. So use it. It's an open book test, so you can use all of your materials, both exam and practical. So make use of that. Make it nice and easy for yourself. I really appreciate you having sticked around. If you joined us a little late, don't worry. You can get all the answers to that part of the study guide that you might have missed. I will upload this video immediately after this session. It takes usually a couple of hours to upload to YouTube. But once it's uploaded there, I insert those links to this review session on our weekly module, and you can find everything that you can possibly want to see from that you missed from that link. But that's all I have for today. I'm happy to answer any questions that you have. If you don't have any questions, I thank you for having joined me tonight. I know it was long. Trust me, I've taken a note who was here, who wasn't. And if you were here, it will count very positive by the time it comes time to do the final grades. If you have any questions, let me know. Otherwise, I wish you a very, very good night, and thank you for joining me again.
Hi, Mrs. Sam. I have a question.
I'll try to help you.
Yes, I think it was the quiz for the lab I was trying to do yesterday, but it just gave me one question, and then I answered that question, and it blocked. It didn't want me to continue, and then I know that I was thinking maybe it was just that one question. At the end, when I came, yes, and then when I came today, I was looking on my grades, and then I looked that it just said like 0.5, which like I missed like a lot of.
No. We'll fix it for you immediately. What I'm gonna do, I'm gonna pause the recording, and we just had a little bit of a break to work through one technical issue that we were having on the Connect. So there was just simply something wasn't loading, and by re-uploading that or re-attempting that site, it loaded again. But that is an indication that sometimes there are issues, technical issues with the software and so on. So if that happens to you, you just drop me a message, and I will definitely work with you. We will figure out what it is, and I will always be glad to give you extension, so you are not going to be in trouble of not being able to complete your work if there's a technical issue. I understand that those things happen all the time, but just let me know and I will work it with you. If needed, we can do screen sharing or whatever helps to figure it out. For me, what's important is that you get a chance to collect all of those points that you are working for, so you get the good grade that you're after. I do ask you to try to stay on top of the workload. Don't let too many weeks fall behind. Like I said, I will give extensions, but I do encourage you to try to submit all of the work on the week that it is due, because very quickly we move on to a new exam, and then you're confused if you're working on an old material and you haven't covered all of the new material and you're already required to take the exam. So I'm here to help you. Let me know sooner you let me know. I can fix it for you. We can work it out together and we can get you those points and can get you to move ahead to the next chapter. So that's all I have for tonight. Like I said, this was a little longer than I had anticipated, so I really appreciate you sticking around with me. And this concludes the review session for the week 4 on the Monday evening. And I will upload this video very shortly overnight. And if you have any questions, as always, don't hesitate to let me know. Thank you for joining me tonight.